Today we're going to talk about carbohydrates, lipids, and proteins. And when we get into carbohydrates, lipids, and proteins, we're really talking about some of the building blocks of life. Life is made out of these different type of molecules, and so we're going to take a closer look at those today. The first thing we need to do is to distinguish between organic and inorganic. And inorganic are compounds that contain carbon. So any compound that contains carbon we refer to as an organic molecule. And these are found in living organisms. We have a nice bird. Inorganic compounds are those that don't contain carbon. There are a few exceptions, as there are in most cases of biology and science. There are always a few exceptions. And in this case, some examples would be like carbon dioxide, carbonates, hydrogen carbonates. These are molecules that do contain carbon, but they're not organic, and so these are inorganic. The first molecule that we're going to look at is called an amino acid. An amino acid is a building block of a protein. We'll take a closer look at these when we get into protein synthesis, but amino acids are the small pieces that are put together to make a protein. There's approximately about 500 known amino acids, but there's 20 that are used in the building blocks of proteins, and those are the 20 that we need in order to survive. Here's our general shape and structure of a single amino acid molecule, and there's a couple different parts to this. The first part would be this amino group. And the amino group is shown right here. I'll draw a circle around it. And so this is the N and 2 hydrogens. Every amino acid has this amino group. It also has a carboxyl group. Here's our carboxyl group. And this is a carbon that's double bonded to an oxygen and then has an OH attached to it. That's our carboxyl group. And the last portion of an amino acid is this R group here. The R group is different for every amino acid. And so of these 20 amino acids, they are all the same in this structure. They have an amino group, they have a carboxyl group, they have a carbon with a hydrogen, and then the last bonding location for the carbon is this R group. And this is different for every different type of amino acid. This is what makes them different. So this is the general structure of an amino acid. The only thing that's making them different is the presence, or what is attached here, is this R group. And let's take a look at some examples. Some examples here. Here we have phenylalanine, and here we have alanine. And so I'm going to actually point out and show where these amino and carboxyl groups are at. Our amino group for the alanine is right here. Here's our nitrogen and 2 hydrogens. On our phenylalanine, it's right here. And the carboxyl group in both of these is a different location. It's on the side right here. Carbon double bonded to an oxygen and an OH. Here's our carboxyl group. And over here, we've got it right there. Oops, just covered up that carbon. And the R group. The R group is what's attached. And so here's our carbon with a hydrogen. Here's our carbon with a hydrogen. And so anything that is also attached to that carbon is the R group. And so this portion right here is the R group. And over here we've got just a pretty simple carbon and CH3. CH3, so a carbon and 3 hydrogens, and that's what the R group is for alanine. And so what is making these different amino acids distinguishable from one another is the presence of their R group that we've highlighted in both of these examples. So let's talk about glucose. It's a type of monosaccharide sugar. Mono meaning one, so it's one sugar, single sugar molecule, and it's found in plants, and it's one of the primary products of photosynthesis. The equation, or the molecular equation for glucose, is C6H12O6. You'll probably want to remember that. It's going to be important when we start talking about photosynthesis and respiration. C6H12O6. In our image over here, this shows us what this actually looks like. We've got carbons in a chain here, or actually they end up being in a branched molecule, and they're connected by an oxygen. And we're going to take a look at glucose, fructose, and lactose, and what makes these different is basically the arrangement, or how these carbon and different elements are arranged within the molecule. So this is glucose. Ribose is another type of sugar, but one of the things that's different about ribose is that it's made of only 5 carbons. It's got a 5 carbon sugar, we go back to glucose for a second, we're looking at 6 carbons. So in glucose, we've got 6 carbons. In ribose, we've got this 5 carbons right here, which makes a big difference. And so we've got 1, 2, 3, 4 carbons in our ring. Our 5th carbon is right here. And then an oxygen again holding the 2 together. Here's another diagram showing us our carbon arrangement in our ribose molecule. One of the key things is it's found in RNA. This is the type of sugar found in RNA. DNA has deoxyribose. In RNA, we just have ribose. So that's one of its key uses or functions. And its chemical equation is C5H10O5. It's a little bit different than glucose. Next, we're going to talk about a fatty acid. And a carboxylic acid with a long tail is what makes up a fatty acid. The tail is either saturated or unsaturated. So here we've got a saturated fatty acid tail, and you can see we've got carbons, and it's got lots and lots of hydrogens. It's saturated with lots of hydrogens. The difference between saturated and unsaturated is that in our unsaturated, we've got some double bonds. So here's some double bonds right here. Here's one. Here's another one. Oops. Here's another one. And in these double bonds, what they're doing is they're removing the ability of these different carbon atoms to make more bonds. And so because of the double bonds, we have less hydrogens. And so as a result of that, we call it unsaturated. So these are unsaturated fatty acids. Glycerol is an example of a fatty acid, and we'll take a closer look at fatty acids and fats a little bit more coming up here. An example of a monosaccharide is that it's a source of fuel, and it can be converted to other molecules or combined into polymers. So monosaccharide is a source of fuel. Some specific examples include glucose, fructose, and galactose. And so here is a nice image that shows us the differences between these three. If you'll notice, if you actually count up the number of carbons, glucose has got 1, 2, 3, 4, 5, 6. Galactose has 1, 2, 3, 4, 5, 6. I'll bet fructose has the same, 1, 2, 3, 4, 5, 6. All of these have the same amount of carbons, oxygens, and hydrogens. The thing that makes them different is their arrangement. And if you notice here, they've highlighted these in red. Fructose has got this portion right here. Its arrangement is different than galactose and glucose. Glucose and galactose are very similar. Just the location of the hydroxide, the OH, has been flipped or changed on both of these, and so that's what's distinguishing or making these different. A disaccharide is also a source of fuel, and it can be converted to other molecules or combined into polymers. And some examples of that include maltose, lactose, and sucrose. Disaccharide means two. And so when we're talking about disaccharides, this means that we've put 2 monosaccharides together. 2 monosaccharides have been put together. So sucrose is made of a glucose and a fructose molecule. Lactose, the sugar that's found in milk, is made of a galactose and a glucose molecule. And maltose is made of 2 glucose molecules. And so how these are actually put together, how 2 monosaccharides are put together, are called dehydration and hydrolysis. We're going to take a look at those reactions in our next video, the second part of our proteins, lipids, and carbohydrates video. If we put disaccharides together, we get something called a polysaccharide. And a polysaccharide has some very specific functions. Some examples include starch, glycogen, and cellulose. They're used in plant cells and cell walls. They store energy. They can also be used as exoskeletons in some organisms. And you'll notice in our diagram here that starch, cellulose, glycogen, they're all much larger molecules. And that's because we've got multiple monosaccharides or disaccharides put together in order to make these larger molecules. So even looking at the root word polysaccharide here, poly means many. So we've got many saccharides being put together, many of these sugars put together to make a starch, a glycogen, or a cellulose. So let's talk about some functions of carbohydrates in animals. The first would be glucose, and this is used primarily as an energy source. It's a very small molecule. It's a monosaccharide. It's very easily absorbed. It's also one of the primary sugars that's produced in photosynthesis. Lactose would be a disaccharide, and it's also used as an energy source. It can be broken down into a monosaccharide, and as I mentioned earlier, it's the sugar found in milk, and so it's very helpful, and it's used to nurse young organisms, young animals. Glycogen, polysaccharide, it's an insoluble energy storage, and it's used in plants and fungi. And it's primarily found in muscle and liver cells in animals um, because it's insoluble in water. Some carbohydrate functions in plants, starting with a monosaccharide. Fructose is a type of sugar that has a very nice sweet taste. And so by some plants producing berries or fruit that has a sweet taste, it attracts animals. Animals eat those seeds, and then they migrate or move somewhere else. They release those seeds, which allow the plant to spread. It's one of the natural adaptations that the plant animal have adapted together. Sucrose, looking at a disaccharide, also used as an energy source for plant. It's unreactive and soluble, which allows it to be moved around the plant. The plant can actually move the sucrose energy around to different portions that need that energy. And lastly, cellulose polysaccharide. It's very large and insoluble, and this is one of the molecules that's used to make up the plant cell wall. A couple different functions of lipids. Lipid is a type of fat, basically is fat. And so one of the most common forms of functions for lipids is energy storage. And we're talking long-term energy storage here. If you were to go and eat some sugar or some candy, it's made of carbohydrates. That sugar or candy is going to be used up very quickly, hopefully if you're active. If not, it's going to get turned into fat. That long-term energy storage, a lipid, it's more efficient than carbohydrates. And so you see animals like a polar bear, they build up their fat reserves over the course of the summer so that they have energy, sugar available for them during the winter when there's less food available. Oils in plants and fish, fats in animals are some examples of that. It can also be helpful in thermal insulation. The fat under the skin provides protection against heat loss. You think about, again, polar bears or maybe sea lions. These organisms have blubber. It helps to insulate and keep their body warm in cold, extreme temperatures. Lastly, buoyancy, also applicable to our polar bear example. Lipids are less dense than water, so it helps animals and organisms that have lots of fat. It helps them to float in water. Some functions of proteins, we're going to look at a couple different ones. Enzyme to start. Proteins can serve as enzymes, and this allows selective acceleration of some chemical reactions. So chemical reactions that are occurring in the cells, they can be sped up by enzymes, and they are essential to these actions actually taking place. A good example of this is digestive enzymes that catalyze the hydrolysis or the breakdown of food molecules. Enzymes are specific to their specific function, and they have an active site, like right here. These substrate move towards the active site, they bond with the enzyme, and then some sort of product is released. And we'll take a very much closer look at enzymes and their functions during this unit. Proteins can also be used as receptors. Protein receptors recognize signals released from other cells and react based on those signals. A good example of this is like in nerve cells. Those receptor proteins are recognizing a signal from a nearby cell, and so that signal is passed from one nerve cell to another, and it's basically relaying that message from maybe your fingertips or your toes or wherever that signal is originating from. It's going towards your brain. Receptor proteins help to process that actual transfer of information. Two other functions of proteins. A structural function would be like keratin, is the protein of hair, horns, and feathers. Collagen provides a fiber network in animal connective tissue. And so here's an example of collagen. Here's a single collagen fiber, and if multiple or lots of these are put together, we can see in this image right here, we've got 1, 2, 3, 4, 5, 6, 7, 7 of these fibers put together, and so we get a nice, really strong, rigid structure. This would be the secondary bundle. We've got our primary bundle here, and so we get this very thick tendon, and this is one of the forms of tendons in animal tissues. Our last example is a contractile and motor, and so proteins responsible for the movement of cilia and flagella in some eukaryotic cells. Actin and myosin are a great example in the contraction of muscles, and so the proteins are actually helping those muscles to move. This occurs very quickly and its energy is required, but it's allowing those muscle cells to actually move. In the second part of our carbohydrates, lipids, and proteins, we'll look at some more of the functions and start comparing and contrasting some of the different types and forms of energy that the different molecules provide.